This is The Development of Twins by Devin Grest, Nicholas Miller, Amber Gottfried, and Abigail Raymond. So an overview of twin development. There are two types of twins, monozygotic and dizygotic. Monozygotic twins are also known as identical twins. They develop from one sperm fertilizing a single egg, and they occur in about 1 in 250 births. Dizygotic twins, also known as fraternal twins, develop from two separate sperm fertilizing two separate eggs. This is the most common type of twin, and if the mother is also a dizygotic twin, the occurrence can be as high as 1 in 17 births. Specifics on dizygotic twins: they develop simultaneously in the uterus. Again, it's two different sperm that fertilize two different eggs, and both of these eggs adhere to the uterine lining, and each zygote develops its own chorion, amnion, placenta, and other developmental organs that assist each individual fetus in development. Monozygotic twins are a little different. They develop when a single zygote splits into two, and the separation of early blastomeres or inner cell masses into different regions of the same blastocyst allow for the separation of the embryo. So they're classified by the number of structures that they share. Monochorionic or monoamniotic twins share a common chorion and a common amnion. Monochorionic diamniotic twins share a common chorion but have separate amnions, and dichorionic diamniotic twins have separate chorions and separate amnions. Now we'll talk about fertilization a little bit. This is what happens when a sperm penetrates the egg and the sperm and egg pronuclei bind. After this first sperm enters the egg, in mammals, there's a slow walk to polyspermy, which is multiple sperm binding, and this is accomplished through the cortical granule reaction when there's an influx in calcium ions. So roughly 15,000 cortical granules are released that prevent the binding of additional sperm. They release proteins such as cortical granule serine protease, and this protein cleaves protein posts that hold the vitelline envelope to the cell membrane, and it clips binding receptors. So when the sperm is bound, several reactions are triggered, including restoration of the cell cycle, the hyaline layer formation, and membrane biosynthesis. These reactions help the egg finish the meiotic divisions required for egg activation. Now we're going to talk about the monozygotic twin development, the pre-trophoblastic split development pathway. So the first of three possible splits occurs before trophoblast formation. In the picture on the left, the two-cell embryo is preparing to split. Here we have two separate but identical blastomeres. So now the separate but identical blastomeres are shown. Now, at this point, each blastomere develops separately with its own inner cell mass and trophoblasts. The embryos develop all their structures separately, as was mentioned in previous slides. So they have their own embryo, their own yolk sac, their own amnion, and they develop within their own trophoblasts. The chorion and the chorionic villi on the superior surface will give rise to the placenta. So at this stage in the pre-trophoblastic split, the identical chorions and amnions have formed, and they've adhered to the uterine lining. Again, 33% of monozygotic twins develop in this manner with completely separate embryonic structures. This is the post-trophoblastic split. So here the two-cell embryo divides and forms a singular blastomere, and the blastomere of the inner cell mass splits after the formation of the trophoblast. So there's separate inner cell masses, but they share a single trophoblast. You can see the two separate inner cell masses within the singular trophoblast. So here, the inner cell masses specify and differentiate into separate and complete structures, including the embryo, amnion, and yolk sacs. the singular trophoblast has begun development of the common chorion and the chorionic villi on the superior surface of the chorion. There's only one chorion here, as compared to the separate chorions in the pre-trophoblastic split. So this, at this stage, it's attached to the uterine wall. There's a single chorion, 2 amnions, and 2 embryos here. The separation of the embryo after chorion formation on day 5 and before amnion formation confirms that this is monochorionic diamniotic twin development. Now we're going to talk about the post-amniotic split pathway. Again, the embryo divides and forms a blastomere. Here there is a single blastocyst and the inner cell mass that remains a cohesive unit, and the trophoblast does not split. So the inner cell mass of the embryo specifies and differentiates into the tissues of the embryo, yolk sac, and amnion, and the trophoblast continues with the development of the chorion and gives rise to the chorionic villi. Again, this forms the placenta. So now this structure has adhered to the uterine lining. There's a single chorion, a single amnion, but there are 2 embryos. So the formation of the chorion and the amnion having already occurred before the division of the embryo confirms that this is monochorionic, monoamniotic, monozygotic twin development. Now we're going to talk about fraternal twins. So fraternal twins have completely separate development. It's 2 eggs fertilized by 2 sperm, and they can be the same sex or they can be different sexes. So in the figures on the left, there are 2 blastocysts forming. And now here, each cell mass has their own separate trophoblast, their own inner cell mass, and they are each their own developing structures. So each embryo, amnion, and yolk sac is individual to each fetus, and both of these cells adhere to the uterine wall and develop into 2 separate embryos, each with their own individual structures to assist them in development, like the yolk sac and the amnion and the chorion. Here you can see the implantation of both of these embryos within the uterus. So in conclusion, mono and dizygotic twins develop in 2 separate but similar manners. And the development of monozygotic twins occurs when one fertilized egg splits into 2. And as we mentioned before, there are those 3 separate ways you can do it: pre-trophoblastic, post-trophoblastic, or post-amniotic. And then dizygotic twins, also known as fraternal twins, develop from the fertilization of 2 different eggs.